and welcome to our lesson number 3, sample and sampling techniques. As you know that chemical analysis forms the basic part in pharmaceutical sciences, but there is one another thing, that is sample. Whenever you are performing pharmaceutical analysis, you will have to take some samples in order to subject them to the analysis. So the sample and the sampling methods are very important, and they are very crucial for doing analysis. So in this chapter we will learn about how to take the sample and what are different sampling techniques. So let's start the video. Lesson number 3, sample preparation and sampling. So dear students, in the last two chapters we have studied about the introduction of pharmaceutical analysis, and we have also studied the basic techniques. Now in this topic we are going to learn that when a substance is under analysis, when it is subjected to analysis, there is something, there is a part of the substance that is taken under the analysis. Most of the time we don't take the whole substance for analysis, we just take a part of it that represents the whole in itself. That process is known as sampling, and the part which is taken is known as the sample. So we first need to understand the importance of sampling. Now as you can see that sampling is a very critical step. It's a very important step because a lot of things, even the result of the analysis also depends upon how you perform the sampling. So it's a very critical step in the analytical process, and you can see that it directly have an influence on the accuracy and reliability of the results. So obviously if something is affecting the accuracy and the reliability of the results, that means that process is extremely important. So that is why in this lesson number 3 you are studying sampling and the ways to prepare the sample. Now how do we take a sample? We take a sample by selecting a representative portion of the larger material. See, for example, you are having a powder and you have to take a sample from it. Then you will take a certain amount from it. Say, let us take it 1 gram, and then you will analyze it. But first you should understand that this 1 gram should represent the whole of the sample. It must contain all of the contents of the sample, although in a small amount. So that is what a sample is, and that is how a sample is defined. Now when you are taking, again when you are taking the sample, you have to ensure that the portion, that means the sample which is being analyzed, it should be the representative of the entire material. Okay, now sampling is very important because it affects the quality of the analytical data that you are getting after analysis, and sampling also affects the validity of the conclusions. That means after analysis you are drawing out some conclusion on the basis of the data that you are getting, and a proper sampling validates this thing that your data is correct, then your data is reliable, and even that your conclusions are true. Now sampling is also critical, also important for obtaining the reliable information about the characteristics of the material. Obviously the material which is subjected to analysis will be having some characteristics. By doing proper sampling, you are going to understand what material is composed of and how its sample should be taken. Now coming to the methods of sampling, there are different methods of sampling, but in a broader way sampling is categorized into these following headings. Number one is random sampling. Random sampling means you are just taking the sample from any part. There is no predetermined order, there is no pattern. You are just randomly and you are just taking that sample from the main matrix. Now the stratified sampling means you have categorized the material into distinct strata, or you can say that you have categorized your sample into different categories. And now when you have defined it into different categories, you are taking the sample from each of the category. That is how you are performing the stratified sampling. Now systematic sampling is the sampling which is done at the regular interval. There is a systematic pattern. For example, you are performing a column chromatography of a substance. Then you will be taking the sample time to time within a fixed interval, so that you can understand that when we were doing column chromatography and we were separating the components, then how and which component was eluting first. Now, how will you prepare the sample? For previously you have studied about how you will take the sample. Now when you have taken the sample, then how will you prepare it so that it can be subjected to analysis? Now this is a very important thing, that sometimes sample cannot be taken as a raw material directly. You first have to convert it into some another thing. You have to complete it, or you have to convert it into a suitable physical form. Then only you can do the analysis process. Now in the sample preparation technique, the first technique is homogenization. Homogenization is nothing but you are uniformly distributing all of the components in the sample, so that whenever you are taking a small portion of the sample, it is the representative of the entire thing. Like in homogenization, for example, there is a powder, and that is and there is a drug contained into that powder. Before taking, you want to know the amount of the drug into that powder sample. Now what you will do, you will first homogenize it, you will first mix the powder properly, so that the whole drug is distributed uniformly into that powder, and then you will be taking a small amount of it. Only in that way this small amount will represent the whole sample. Now the second process is extractions. Sometimes we remove the analyte of interest from the matrix. Like for example, we want to test what are the chemical constituents in a plant, or say let's say in a leaf of that plant. Now you don't have to take the leaf, you just will take the extract from the leaf. By performing some extraction procedures, you will take the extract from the leaf and then you will analyze. What will be the benefit of this process? The benefit of this process is that when you are taking the extract from this, you are leaving the rest of the matrix, like leaf, its chlorophyll, its other organic part, because these organic part are not required for our analytical procedure. We are just interested in the chemical constituent, so the best way to do is to get the extract. Next sample preparation is filtration. Dear students, you have already seen filtration. You see that filtration is nothing but we are separating some solid particles from the liquid samples. So let us say that some amount of solid is mixed with the liquid. There is a mixture, and it is actually not a homogeneous, it's a heterogeneous kind of mixture, and you are using a funnel, and you are separating that solid from the liquid. You will pass, there will be a filter paper in the funnel, you will pass that mixture from the funnel, and the solid particles will get retained into that funnel, and the liquid will come in the beaker or in the conical flask. Now either you can analyze solid or you can analyze this liquid. You have separated these two things and you are analyzing them separately. So that's how we use filtration. Now there are also some additional processes for the sample preparation. The previous processes were the most common processes that we use in our day-to-day analysis, and it is used most commonly in all kind of analysis. But there are some special processes that are also very important to know. The first process is digestion. Digestion is nothing, but actually it happens during a kind of analysis known as like gravimetric analysis. While you are performing gravimetric analysis, you do this kind of sample preparation. Now what we do in gravimetric analysis, see, we form a precipitate. There is a precipitate which is being formed in the sample. Now we keep this sample hot and we allow it to stand by for, say, let's 2 hours or 1 hour or 3 hour. And now what will happen, that small particles will get dissolved and it will form a larger particles, and this large particle will get accumulated on the precipitate. So there will be a breaking down of something, and obviously there a larger thing is formed. This is known as the digestion process. So digestion process is very important in the gravimetric analysis. Obviously it is not used frequently. It is mostly used when we are dealing with the precipitates. Now next method is derivatization. Derivatization is like, for example, in a chemical analysis, we have reacted two substances A and B and we have formed a new substance C. Now the thing is that this C is, let us say, it is unstable. Now we will convert this C into a stabler form, which is D. Now this process converting C into D, which is more stable, is known as derivatization. Derivatization converts the sample into more suitable or more understandable form, or you can say that in that form which is easier to analyze. So this D has some characteristics which makes it a better analyte than the C. So this is what derivatization is. Now coming to the dilution. Most of the time we also use dilution. Dilution, simply it reduces the concentration for analysis. Dilution means you are increasing the amount of solvent. You are increasing the amount of solvent in comparison to the solute. So these are also required in some different kind of analysis. Now let us understand why it is important to make a proper sample preparation. Why proper sampling is required, or proper sampling preparation is required. So the first thing is that it increases the accuracy and precision of our analytical measurement. Whatever we are doing as analysis, it will obviously help us in doing it. Second thing is, there will be a minimization of the interference and the matrix effect. Obviously when you are extracting out the main component of the sample, like for example in extraction, I have told you that you are taking an extract of something and that extract contains the material which you actually require. Like for example, there is a leaf, you don't need the leaf, you need only the chemical constituent which is present in the leaf. So you have extracted that leaf, you have taken that chemical constituent into some solvent or some vehicle like ethanol. Now you are focusing on only on that chemical constituent. There is no, you don't want the other parts of the leaf, other anatomical parts of the leaf to interfere in your analysis. So that is why you have taken out all the extract. So this helps us to understand why we want to minimize interference. So that is why we are performing a sampling technique. Now there is an improved reproducibility of the result. See, analysis is always considered good when the results are reproducible. If the results are not reproducible, you cannot rely on that analysis, because reproducibility directly says that how much precise your experiment was, how much you can rely on your experiment. So a proper sampling techniques also helps us to gain the reproducibility while performing experiment. So this was all about sample. So let us take the summary of it. Number one, it is sampling is very important for obtaining the representative portion of material for analysis. We don't have to take all of the content, we just have to take a small part, that is known as sample, and that part will represent the whole matrix. Second thing, there are different sampling methods. We have studied different sampling method like random sampling, stratified sampling, systematic sampling, and by these way we are by these different sampling methods we are addressing different kind of problems in during the analytical procedure. Now there are some special sample preparation techniques. Some are very common like homogenization, extraction, filtration. Others are a little bit lesser used but still they are important like digestion. Now the last thing is proper sample preparation is extremely important, as you have all understood from this chapter, and if we are doing the proper sampling we are obtaining meaningful data and we can also draw the valid conclusion. Thank you. So dear students, I hope you have understood the sample and different sampling techniques. This is a very crucial part as I have told earlier. Now we will move on to our lecture number 4. Our lesson number 4 is the quantitative analysis in pharmaceutical chemistry. We will understand what quantitative analyses are. So we'll meet in the next video for the lesson number 4. Till then, goodbye.